Okay, so actually, from this last talk, there couldn't be a better segue. Kim introduces the intestine incredibly in a nice manner, and as well, getting into the 2D monolayers that he's talking about is exactly what I'm going to go into through most of this talk. So it's a little bit different. We try not to ask a specific scientific question, but characterize a tool so that all of you can ask the questions as we just saw from this last study. Yeah. Okay, so I showed this slide here talking about the composition of a stem cell niche. Kim really nicely introduces the intestine. I like to point out strictly, it's a very unique epithelium. Most of the epithelial tubes remain as tubes, and the stem cells don't isolate to a particular spot in that tube. And the point we heard over and over is it's not the stem cells themselves, but their location that determines the fate. So the intestine's the only epithelium that has evolved to establish these crypts. Right, the very, very base here, and we figured this is just due to the harsh environment and the massive turnover. Kim talked about numbers. I'm going to more focus on the timing of it, right? You're really replenishing this quite a bit. We know exactly where they sit. The stem cells intercalate between these granular Pannis cells, and this is where a lot of the signaling has been described. In Hans's lab and many others sum up quite a lot of work in these signaling pathways. So we know, if you see the Pannis cell here shown in blue, the stem cell shown in yellow here, we have key signaling pathways, EGF activating RAS-RAF signaling, Notch delta, where the notch becomes active, both the canonical and non-canonical Wnt activating frizzled and LPR5/6 receptors. We also get signals from the stroma in our spondin, again activating Wnt, and an inhibition of BMP. And this is what maintains the stemness of the stem cell. So again, going back to what we've just heard a lot of, it's really the environment that keeps these stem cells as stem cells. So this little model right here came where at the very base of the crypt, they encounter high Wnt signaling, high EGF, and an inhibition of BMP. So the reason I show you this slide is this is how the original organoid media was founded. So we all know a group of Hans Clevers, an extremely talented postdoc, Toshiro Sato, who now has his own lab in Japan, came up with this idea that if we know what keeps a stem cell as a stem cell, we can create a media in an extracellular matrix through matrigel, so the signals can be transduced, in a way where the media replicates the stem cell niche. So this is the concept that we take in designing all the organoid media stem cell. So what they were able to show in a transgenic mouse, the LGR5 GFP mouse, so the stem cells are all marked in green. When you culture the mouse crypts, as seen here, place them in a matrigel dome, add the media, again recapitulating the stem cell niche, you immediately get these crypts closing up, right? They want to form this cystic structure. Within a couple days, what you'll see is you'll get this budding structure, again, indicative of the high Wnt signaling, which is pushing out these crypts. They characterize this really well, looking at the key markers of the intestine, and they find by staining for villen, representing enterocytes, you get a very nice apical localization, indicating the lumen is maintained. When we look at the functional differentiated cells, looking at mucin 2 for goblet cells, lysozyme for Pannis cells, which localize superficially on the organoids, and then small amounts of chromogranin A, indicating the secretory enteroendocrine cells. All the cell types are there. So this model in the mouse here really, really recapitulates the in vivo intestine. So at STEMCELL, we partnered with the Hub, which is the company that came from the Clevers group, and we also partnered with the Cincinnati Children's Hospital, which basically did the same thing through ES and IPS lines. And we're able to grow the intestinal organoids, which you've seen in the last presentation, and these are what our ES-generated intestinal lines look like. So there's a few differences. The key one, and they address different questions. The key one is the adult-derived intestinal stem cells are strictly an epithelium. You can ask questions based on the epithelia. So what do you want to look at in the intestine with no external influence? The ES-based intestinal organoids, they have a mesenchymal component. So this you get a system where the mesenchyme is actually providing some of the signals to the intestine, and you're addressing a little more complex structure. Again, both very useful in different situations. And we obviously have media that support both of those, the IntestiCult, which you heard about, and our STEMDiff intestinal. So again, I told you I was going to talk about the two-dimensional monolayers, and I came to introduce the CACO2 cells, which is a tumorigenic colon cancer line, which has been around forever. I don't want to discredit a line like this. Many discoveries, many of the drugs that are currently used in any kind of intestinal abnormalities have come from these CACO2 cells. But now we have a 3D organoid. So this is now a more complex structure. We have the key differentiated cell types, we have intestinal stem cells that are constantly proliferating, and this gives you a much more relevant structure. And then now we've seen the 2D monolayer as well. So one of my group put up this cartoon on my desk and says, why do we want to go backwards? Right? Now that we have 3D, why do we want to go back into 2D? And I think you got a really nice example of that in the last talk. We're not really going backwards. We're taking a step forwards and introducing new applications that we couldn't have got on before. So I'll just summarize these systems really quickly. Right? The immortalized cell lines, such as the CACO2 cells, they're cheap, they're easy, they grow in serum, they always grow. We don't have the variability we talked about before. So very consistent, but the disadvantage is they're not very representative. They're a tumor model. These divide because they've picked up mutations that immortalize these cell lines. We've talked about the 3D organoids. They're absolutely more physiologically relevant. I think every week I get a new publication that really pushes the envelope on new questions we can ask with this model. The disadvantage is it's complex and requires an extracellular matrix in the form of matrigel or basement membrane extract, which is a little bit more variable. And we had questions too on the variability patient to patient. And then you get to the 2D monolayer, and you combine the relevance that you see in 3D in the convenience of the 2D. So you lose the 3D physiology, but you get this more manipulable system. I want to point out here too, the reason the organoids are a really key advancement is you can't take the patient biopsies and go straight to monolayers and keep them going. And this is strictly because there's not enough stem cells in these biopsies that you can get a density enough to establish these sheets. So the organoid technology provides this step where you can actually advance and increase the number of stem cells, and that seeding onto the monolayers is only made possible by this organoid step, right? So it's really a stepwise process that you have to take to establish these. So I want to talk about some key characteristics in these monolayers, and we'll provide, again, instead of asking a scientific question, we'll provide a characterization that hopefully helps you on your characterization. So we'll talk about cell polarization, the formation of tight junctions, the appearance of these differentiated cell types, which is absolutely key. We don't want to look at a monolayer of stem cells. We'll look at the epithelial barrier properties, and then finally the functionality of ion transport. So when we look at cell polarization and we compare the CACO2 lines and the monolayers, and in brackets is the day that we're looking at. So the basic protocols are a 21-day-old CACO2 line is the functional one. This is industry standard. This is where you want to be looking at. The organoid-derived monolayers form in 7 days. And if you look at these images, what we're looking at is a top view coming in, and then you can look at the cross-section here on the bottom. We stain for villi, marking the apical villi of the intestine, and then we stain for ecadherin. And what you want is consistency in this sheet, right? So you want to see, it's okay to have ups and downs in the plane, but when you look at the superficial view, you don't want to see junctions here shown by ecadherin and sections in the same plane that don't have these junctions. Right? When we look at the organoid-derived monolayers, you can see within the plane you get very consistent junctions, and this will hold true in the functionality of these organoids, which I'll show later. When we look at specifically these tight junctions, looking at key markers which localize between the cells, we can see E-cadherin, cytokeratin, and claudin-4. And what we see from this slide is within each complex, such as E-cadherin and the claudin, you can see that the same plane is maintained. When you come more superficial, where the cytokeratin is, we see that same consistency within the plane. So the tight junctions are there, the integrity of the monolayer is going to be maintained because of these junctions. We also have a unique protein, relatively unstudied, called tricellular, which is a tricellular junction. So at the adjoining point of these epithelial cells, you'll have where where the trifecta is, you'll get a protein that localizes, and it's been characterized that's absolutely necessary in some type of intercell signaling. So although the function of it is still being done, you can see when you compare the CACO2 cells, again, you get very spurious localization of this tricellular. Yet when we look at the organoid-derived monolayers, it's really in every single point where each cell forms. Right, so this again is demonstrating more relevance within this organoid-derived monolayers. When we look at cell differentiation, and I think Kim showed you this a little bit nicer than I did, especially with the sequence data. We look at staining for mucin 2 representing the goblet cells, and then EpCAM again showing the junctions. And you can see small amounts of goblet cells, which is very consistent with what you'll see in the endogenous intestine. And this is very important because you'll actually get the mucus produced and sitting on top of the monolayer. So moving forward to when you're looking at co-cultures, which are possible with this, it's very clear that you're going to need the goblet cells for this immune function. And then you can see just the cross-section. They're actually secreting mucus from these cells. So now we turn a little more to the functionality, and we look at the quality of the epithelial barrier. So again, I've been showing you these junctional proteins and talking about the integrity of these cells, right? You really want them stuck together. The way the industry standard that used to measure this is transepithelial electrical resistance, or tear measurements. And what you're really assessing is the continuity, so how well these cells are held together, and then also the width, which comes into a factor, right? You're just looking at resistance between the apical and basal parts of the cell. I want to point out too, this is currently one of the strongest features of CACO2 cells, right? This is something you really can do with this immortalized cell lines. So the values they're getting here is what we're comparing our measurements to. So when we look at CACO2 cells, just grown in 10% FBS, and then monolayers and intesticle, or grown in testicle and rock. After this, we switch to MEM plus 2% FBS for the actual study. And when we compare these, we get an average tier value of about 300, which is well above what we need to do any of the measurements here. I'd also like to point out too, you get a little bit more variability in these human colon monolayers, and I think that this gets back to one of the questions that was posed earlier. It says, how do you deal with this variability? You can deal with this variability and just say it's, unfortunately, it's a product of the system, or you can take it the other way and say, we're looking at individual patients here. We expect that these are different, right? So you're actually getting accurate measurements for the individual. And this is taking the approach of more precision-based medicine, right? If we're looking for an accurate depiction of how the integrity of these cells, we expect there to be some variability. We then made an experiment where instead of addressing the integrity, we're looking at the permeability of these pathways. So we saw before from the images where we stained for the proteins which we know should hold these cells together, and we found in the CACO2 cells it wasn't completely there. We now test this functionally by taking labeled dextran beads, adding them to the apical portion of this transwell, and then making a measurement to see how much has actually gone through and passed into the bottom. With the integrity and the tier values which we expected, you shouldn't expect so much coming through. Yet when we look at this over a time course, at 0 hours we get about the same, 3 hours similar, yet at 24 hours in the CACO2 bottom well, we've seen a lot of these beads have passed through. And although we know that the permeability, the tear value of the cacotubes is solid, what we're forgetting is these are a tumor line. They're actively filled with stem cells, and these stem cells are dividing very rapidly. And I think through these divisions, they're actually opening up pathways for the dextran beads to go through. So when we're looking at functional studies, looking at what comes from the luminal side of the intestine and can be pulled out, we're not getting an accurate depiction here in the cacotube cells. So this opens a new frame of study from the organoid-based monolayers here. This is just what I said. Yeah. Okay, and then finally, the last slide I'm going to talk about functionality here. The intestine is an important regulator of fluid ion balance, and this goes through a bunch of key receptors, right? Sodium channels, CFTR, which we know regulates chloride, and calcium channels are sort of the key components of the intestine. And this is pretty, we also know too that any abnormalities within these transporters leads to diseases, right? So cystic fibrosis, diarrhea, and any type of chloride imbalance. So the way we test this is with a Neusing chamber, and this is taking a short circuit current, which measures the electrical potential across the cell membrane as these ions are transported. So when we look at this is a mouse monolayer, and we look at in either inhibitors or activators of these key channels. So looking at inhibitors of sodium, activators, potentiators, and inhibitors of CFTR, and an activator of a calcium channel, what we see from the measurement is we see a small drop. So from this chart, what we're looking for is a difference in potential after giving the drug. So we look at a small drop after giving the sodium. We see a massive increase after giving forskolin, the CFTR activator, a small divot through the potentiator, and then this big drop again through a CFTR inhibition, and then finally a huge peak for the calcium activator. So what this demonstrates is you actually have a very responsive monolayer. Not only is it consistent, it's going to respond and function as the intestine would through the various ion channels. When we make comparisons here, looking at a couple of donor samples, so human colon now, two different ones to look at variability, we can see, looking at strictly the forskolin, so this is a CFTR activation, we see a huge difference in both the two human samples as well as the mouse colon, which is not seen through the CACO2 lines. Again, we try the 21-day time point, but we also compare the 7 days that we look at in the organoid-derived monolayers, just to make sure the timing is not essential here. When we look in the inhibition of CFTR, we see something very similar. We lose a little bit in the mouse, but the human organoid-derived monolayers respond very well. And again, very little response in the CACO2 cells. So again, we're looking at physiological relevance. We really don't see this in the immortalized cell line, and the organoid-derived monolayers really give this opportunity to ask questions on the specific diseases we didn't see before. So I hope I've given you a little insight here and shown you that organoids, I think we knew that organoids provide the opportunity to establish monolayers. Kim showed this very nicely. These monolayers really give you nice access to both apical and luminal compartments. So we saw the microbial study that Kim's talking about, where you get the apical side, but you can also go in anything from the luminal layer as well, or look at transport across. These monolayers are clearly more physiologically relevant, and the timing that these monolayers establish in 2 to 3 days and are ready for assays at 7 days, saving you a couple weeks on these immortalized cell lines. So with that, I'd just like to acknowledge the team. This work was mainly the work of Martin Stahl in Vancouver, but support from the whole group there, and then the group, many are who in the audience here at the Cambridge site, Roxana Salvatore, Jose, and Tom. Funding, part of this project is through the Intense grant, and MyTex is a company in North America that provides us with postdocs. I do want to take one more second here and talk about one slide, as well as showing a whole bunch of beautiful pictures. The organoids have really clearly made a difference, and it won Nature's Method of the Year in 2017. So as much as the Method of the Year is just something that happens, I'd like to point out the timing. So 2017, just a couple years ago, there's a couple other things that won this award. CRISPR-Cas gene editing won in 2009, and single-cell sequencing won this award in 2011. So if you look at every paper right now, we were at a meeting yesterday where almost everybody made the joke, you can't publish anything without single-cell seq. But you look at the transition, where it's about 7 to 9 years that actually comes from the method of the year to what's actively used. And you think, we're only 2 years off this. If you're not working with organoids yet, you can, and you're still at the absolute forefront of the technology. So you can get way ahead. I also just want to point out that as stem cell technology, we have the Organoid Information Hub, we have videos on how to do things, we have webinars, talks, these talks will be online, and we also have tech support. Kim was, I think, kind of joking, saying he can phone me and say, I need help on this. This isn't exclusive to Kim. This is everybody who buys the products. We really, really want to push the science forward. So, sorry, Kim. And this is what we do, and the tech support people are incredibly trained, and if there's anything we can't help, and there's various levels. They'll go to the intestinal team, and somebody will find you some support there. So guys, with that, I want to thank you for your attention, and take any questions.